Neuronal noise or neural noise refers to the random intrinsic electrical fluctuations within neuronal networks. These fluctuations are not associated with encoding a response to internal or external stimuli and can be from 1 to 2 orders of magnitude. Most noise commonly occurs below a voltage threshold that is needed for an action potential to occur, but sometimes it can be present in the form of an action potential. For example, stochastic oscillations in pacemaker neurons in suprachiasmatic nucleus are partially responsible for the organization of circadian rhythms. Neuronal activity at the microscopic level has a stochastic character, with atomic collisions and agitation, that may be termed noise. While it isn't clear on what theoretical basis neuronal responses involved in perceptual processes can be segregated into a neuronal noise versus a signal component, and how such a proposed dichotomy could be corroborated empirically, a number of computational models incorporating a noise term have been constructed. Single neurons demonstrate different responses to specific neuronal input signals. This is commonly referred to as neural response variability. If a specific input signal is initiated in the dendrites of a neuron, then a hypervariability exists in the number of vesicles released from the axon terminal fiber into the synapse. This characteristic is true for fibers without neural input signals, such as pacemaker neurons, as mentioned previously, and cortical pyramidal neurons that have highly irregular firing pattern. Noise generally hinders neural performance, but recent studies show, in dynamical nonlinear neural networks, this statement does not always hold true. Nonlinear neural networks are a network of complex neurons that have many connections with one another, such as the neuronal systems found within our brains. Comparatively, linear networks are an experimental view of analyzing a neural system by placing neurons in series with each other. Initially, noise in complex computer circuit or neural circuits is thought to slow down and negatively affect the processing power. However, current research suggests that neuronal noise is beneficial to nonlinear or complex neural networks up until optimal value. A theory by Anderson and colleagues supports that neural noise is beneficial. Their theory suggests that noise produced in the visual cortex helps linearize or smooth the threshold of action potentials. Another theory suggests that stochastic noise in a nonlinear network shows a positive relationship between the interconnectivity and noise-like activity. Thus, based on this theory, Patrick Wilkin and colleagues suggest that neuronal noise is the principal factor that limits the capacity of visual short-term memory. Investigators of neural ensembles and those who especially support the theory of distributed processing propose that large neuronal populations effectively decrease noise by averaging out the noise in individual neurons. Some investigators have shown in experiments and in models that neuronal noise is a possible mechanism to facilitate neuronal processing. The presence of neuronal noise, or more specifically synaptic noise, confers to neurons more sensitivity to a broader range of inputs. It can equalize the efficacy of synaptic inputs located at different positions on the neuron, and it can also enable finer temporal discrimination. There are many theories of why noise is apparent in the neuronal networks, but many neurologists are unclear of why they exist. More generally, two types of impacts of neuronal noise can be distinguished. It will either add variability to the neural response, or enable noise-induced dynamical phenomena which cannot be observed in a noise-free system. For instance, channel noise has been shown to induce oscillations in the stochastic Hodgkin-Huxley model. Ions exist inside and outside of the neuron and are subject to many bodily conditions. One major source of noise arises from ions or molecules in response to the third law of thermodynamics. This law states that the entropy of a system approaches a constant value as the temperature approaches zero. Since the body maintains temperatures above 0 K, the molecules are subjected to increased kinetic energy, or motion. The stochastic, or random, movements give rise to receptor noise produced by the constant bombardment of ions, as described by Brownian motion. Ions are constantly being leaked across the membrane in efforts to equalize the ionic gradient produced by ADPase channels embedded in the membrane. These leaky channels permit the movement of ions across the membrane, leading to small fluctuations, or noise, in the membrane potential. Synapses are another major source of neural noise. More than often, there is random exocytosis of vesicles containing neurotransmitters, which eventually bind to the postsynaptic membrane, leading to a spontaneous action potential by graded potentials in the postsynaptic neuron. It is considered the largest amplitude noise source in the cerebral cortex. The external noise paradigm assumes neural noise and speculates that external noise should multiplicatively increase the amount of internal noise in the central nervous system. It is not clear how neural noise is theoretically distinguished from a neural signal. Proponents of this paradigm believe that adding visual or auditory external noise to a stimuli and measure how it affects reaction time or the subject's performance. If performance is more inconsistent than without the noise, the subject is said to have internal noise. As in the case of internal noise, it is not clear on what theoretical grounds researchers distinguish external noise from external signal in terms of the perceptual response of the viewer, which is a response to the stimulus as a whole. Electroencephalogram or EEG can be used to measure the brain since the signal-to-noise ratio is poor, so noise produced by the brain can be detected in vivo. Local field potentials can be used to test the noise present in large neuronal networks. This measure of voltage can be used to determine the interconnectivity by the level of noise. Local recording has contributed much to discovering many of the new sources of ion channel noise. Patch clamp technique was crucial to determine shot noise because the use of intercellular recording was not able to show the movement or detection of the few ions escaping at a time.